this is kind of, I guess, a little bit informal. I was going to make slides, but then decided that instead of me telling you what I think you should know about, transition to independence, I thought I'd tell you a little bit about where I've come from and then leave the floor open to you to ask me about different things, because there's obviously quite a lot to cover. So, as a little bit of a sort of summary of how I got here, I came to Newcastle in 2000 and I think I would have checked this beforehand, 2012, I think, to do a master's degree. So I initially actually was going to go and do a PhD in Edinburgh. So I had a funded PhD by the BBSRC at Edinburgh, was to study, I think it was place cells in the entorhinal cortex of mice. But the problem was that I really wanted to study dementia. This wasn't a project on dementia. It may have been slightly relevant to dementia, but it wasn't really on dementia. So I took a risk. Now, things are a bit different when you're younger than when you get a bit older. Would I have made the same risk now? Possibly not. But so I decided to turn down the funded position and pay to work to do a master's at Newcastle, but because it was on a subject that I wanted to study. So this meant that I spent the summer working in a place called Star Instruments, making thermometers and pressure gauges in a factory. If you ever want to talk about thermometers and pressure gauges, I am your man. I know a lot about them. So I spent a bit of time doing that, and then I came to Newcastle and did my master's. This was a massive risk because I did not have a guaranteed PhD. So I then had to apply frantically for PhDs during the year I was at Newcastle. Then with my last application, as in all of the other irons were out of the fire, I got accepted with the person who was supervising my master's project. So I then started my PhD at Newcastle, which was studying the neuropathology of the visual system in dementia with Lewy bodies. Anyone who knows anything about dementia with Lewy bodies probably knows hallucinations and sort of pareidolias, which are like misrepresentations of visual stimuli, are quite a common feature. Hallucinations are one of the core features. So I worked at that for 4 years in the end. As I was coming to the end of my PhD, I'd come up with a few ideas. So, the take-home message from my thesis is, despite visual symptoms being a really big thing in DLB, the visual system doesn't have very much pathology, which is not a particularly interesting thing to find, but it's, I guess, negative results are still results. We found a lot of molecular changes, but nothing in terms of histopathology, much anyway. But this got me interested, why exactly is the visual system one of the few parts of the brain that never gets Lewy bodies? So this idea that we have, I'm sure most people here are familiar with the concept of prion-like spread, which is where a misfolded protein meets the native protein. It induces it to misfold and sequesters it into aggregates. So one of the things that's necessary for that is you need to have the normal protein present in sufficient quantities to be seeded. So during the end of my PhD, I thought, wouldn't it be interesting if the visual system actually had much less alpha-synuclein? Because that would be a neat explanation for why it never gets Lewy bodies. So I wrote a little grant to AR UK. It was like a pilot project grant. It was funded. We found that the visual system actually doesn't have much alpha-synuclein. The rest of the brain does. So that was kind of interesting. And we used this data then for me to get my first fellowship, which was to look at alpha-synuclein and its association with vulnerability to pathology. And then about a year and a half ago, I got my senior fellowship, which has expanded this work to trying to understand, why does alpha-synuclein accumulate in the first place? Why do risk genes for Lewy body disease, heterozygous variants in, lysosomal proteins, for example, lysosomal genes, are associated with risk of Lewy body dementia, but, in biallelic forms, they cause pediatric diseases. So we've now expanded into pediatric neurodegenerative disorders as well to try and gain insights into what's happening in these conditions. So that's how I've got here so far. In terms of the kind of main challenges, so I would say that independence is a very gradual process along this way. You don't just become independent overnight. There are various stages. So even during a PhD, for example, in my final year, we investigated an idea that I sort of found along the way. It was something that was kind of uniquely mine, which was kind of a nice thing to study. The project, obviously, after my PhD was something that I had conceived. So this is something that's, again, a little bit more independent. But even when you're doing a fellowship, you're still not really independent. You're still dependent on a lot of people. And even now that I've reached a sort of faculty position, which people would consider to be independent, it's extremely naive to think that science is ever an individual pursuit. Science is absolutely a collaborative effort, and you're still very dependent on lots of people, including people who are more advanced than you, that help you understand things, such as the university system, the university's procedures, but also the wider research community as well, because with the way research is going, there is simply no way that one lab, no matter how good they are, no matter if it's the LMB in Cambridge, they still can't do everything themselves. Collaboration is always key. So, yeah, as I said, it changes along the way. The biggest challenges I think that I have faced is kind of a tricky one, and I toy with this a lot. I think you never ever get over the disappointment of things not working. I mean, it gets easier to cope with, but it still is a massive frustration. It still feels like a setback. You don't get to a stage where you're completely immune to setbacks and suddenly, everything's better. And yeah, I'm trying to think what else. Grant rejections, that becomes a big thing. And of course, the pressure changes. The pressure changes from I need to get more funding so that I still have a job to I must get more funding to keep my team employed, which is a very, very different sort of stress. But I've sort of given you an overview of who I am and what I am. But now I'd like to hand the floor over to you so that you can ask me some questions or any discussion that you want to have, that maybe I could share some of what I've learned, which is of course just my opinion, and many people may disagree with it, but it's what I found helpful.
I was wondering if you had any reflections on imposter syndrome, whether it's got worse or better over it, and how you've dealt with it.
I think it's an excellent question. I don't think it's something that ever goes away. I don't think you ever reach a stage where you think, do you know what, I'm amazing, that's great. That just never happens. You always think, have I got here by luck? Was that last finding just a fluky one? And am I actually not cut out for this? It never really changes. You do cope with it better. It's a bit like when you give your first talk. When you give your first talk in front of a big audience, it's really stressful. You're really worried about. Well, certainly I was really worried about it. Sweating, panicking, and very nervous about it. I still get nervous now. Everyone gets nervous before they give a talk, I think. But you just learn to cope with it a bit better. And I think that's probably the case for most things in academia. There's the job insecurity. It never really goes away. Even when you're tenured, nobody's really tenured. I mean, if someone stops publishing papers tomorrow, stops bringing grant income in, I don't think their position in its current form is likely to be tenable at any institution. But that's the same as, if I worked in Sainsbury's, which I did do for several years, and I start refusing to do my job, it's unlikely that I will stay in employment. So I think that again it changes, but I think that you just become more used to coping with it, and that's I think for most things.
Just touching on job security, I wondered if you've ever been interested in moving over to industry and why you didn't do that. It's
a really good question. I probably should have explained. When I was talking about my career to date, it sounds like I really like Newcastle. You may detect from my accent I'm actually not from Newcastle. So it's not necessarily that I just decided to stay at Newcastle and I did everything to stay here. That's not the case at all. I came to Newcastle deliberately because of its sort of the prestigious place that it has in the dementia with Lewy bodies field. And it's kind of something that has continued, as in I've not really had any reason to leave because I have everything I want here. I have a stimulating work environment. I'm surrounded by excellent colleagues. And I think we do science really well. Not so much, I'm not saying in terms of just cold hard metrics, but we're a collaborative group. We work really well together. Everyone has a voice that is valued, and everyone contributes to the discussions from the lowest to the highest level. Lowest is a bit of a loaded term. I mean, across all levels of employment, everyone contributes and it means something. So that's why I've stayed here. I'd say I've never considered the jump to industry because I've enjoyed what I'm doing so much. If that makes sense. As in, I love what I do. I still come into work every day thinking, God, I'm so lucky. And it comes back to imposter syndrome as well. You kind of feel to yourself sometimes, how have I got so lucky to do a job that is essential? It feels like a hobby sometimes, We get to explore interesting things and it benefits other people. I mean, what's not to love? So you do feel really, really lucky. I appreciate that. And luck is a big term as well, really important term. I do not kid myself that I'm something special. I have been extremely lucky through my career. I continue to be extremely lucky. And I think luck counts for a huge amount. It's a bit like the England football team, I guess. You know, would you rather be a good football team or a lucky football team? I think England have largely been quite lucky so far. But I take a lucky team every day of the week over a team that plays beautiful football. So, yeah, that's why I've never made the move to industry. Perhaps if life had handed me different cards, maybe it would have been different. But that's the reason so far.
How did you come up with your first fellowship idea, and what was the process like from like inception to actually submitting your grant for that?
That's a really good question. I might have to think a little bit here. So let me see. I guess it all kind of occurred quite organically and at different times. So my PhD project was on the visual system in DLB. We didn't find much alpha-synuclein pathology. So the first question was, why does it not get much alpha-synuclein pathology? And the reason, or we think, is that it doesn't contain much alpha-synuclein normally. The question from that was then, that's on the sort of regional level. So that was like primary visual cortex versus the rest of the cortex. But what about individual cells? And at around that time, there was a group in Japan whose name escapes me, but they had looked at alpha-synuclein expression across multiple cell types. They found that some cell types always expressed alpha-synuclein and some didn't, in mice anyway. So that then got me interested, because anyone who looks at Lewy body pathology, one of the most interesting things for me anyway, is that there are some cells in the brain that develop Lewy bodies and some that never do. No matter how advanced the disease is, no matter how severe a person has it, those cells will never have Lewy bodies. So the question is why. I'm still interested why. If someone knows, tell me, because I mean, I'm fascinated. And how does this change the phenotype? Because, yeah, I mean, I don't want to go into too much detail, but these were the questions that we were asking, and then that kind of went on and on and on until it was, well, let's actually directly compare pyramidal neurons to interneurons, you know, because interneurons never develop Lewy bodies, but every symptom of DLB suggests that interneurons should be affected. So I always think DLB is very reminiscent in some ways of epilepsy. It's almost like a functional connectopathy rather than a frank neurodegenerative pathology. The reason I say that is because if you look at postmortem brain tissue from DLB, they have virtually no cell loss. You know, apart from the substantia nigra, you know, a couple of thalamic nuclei, some of the monoaminergic nuclei, apart from that, the cell is remarkably preserved. The brain of someone from DLB is no less in weight than a person without dementia. A person with Alzheimer's disease is over one standard deviation below the control. So why is this, if this is such a widespread neurodegeneration, then why is the brain not lighter? And why is there not more obvious cell loss and more obvious atrophy? So all these questions kind of coalesced into a sort of project which was looking at the difference between these different cell types. And as I say, I still find it interesting today. It's just changed a little. Again, everything just changes a little bit. The same things kind of remain throughout, whether it's dealing with, you know, sort of imposter syndrome or dealing with setbacks or whatever else. It kind of always, for me, it's always stayed the same. It just changes a little bit. As in the same things remain, but they change.
So I think a kind of a related question for me. Obviously, you've done very well going from postdoc to junior fellow to senior fellow. I think as an ECI, I don't know about anyone else, I always feel like you support like the amount of things you have to try and balance from getting the work done. You need to start and take student supervision, you need to publish papers, you need to be doing network and raising your profile. How have you been able to balance that, and how have you been able to time that into when you've started developing and developed your next fellowship application? How long does that take? So your senior fellowship, for example, how long did that take for you to actually get all of the things that make you, because the amount of framework that we need to be seen to be doing nowadays?
There's no question, like it's really, really tough. Time management is such a massive issue. I mean, it's probably not a very helpful answer, but sometimes I really don't know. I don't know how it all gets fitted in. I really don't. You're quite correct. You know, it's a constant, you know, yeah, you've published a load of papers, but, you know, what are you doing in terms of mentorship? Or you're doing mentorship, yeah, but where's the grants? You know, it feels a little bit like whack-a-mole, you know, as in like as soon as you address one thing, something else pops up that you should be doing that you aren't. And it's really difficult, and it becomes even more difficult as you start having a family and suddenly, you know, working ridiculous hours and work is no longer you know, a reasonable possibility. It's, yeah, I'd have to think. I honestly don't know sometimes where the time comes from, if that helps. So obviously during my sort of junior fellowship, we made an interesting discovery, and that actually formed the basis of the senior fellowship. So at Newcastle University, we're very fortunate that we get some excellent master's students who come and do a 6-month placement in our laboratories as part of our research master's program. So we also get a lot of junior doctors through the Academic Foundation doctoral program through the Northern Deanery. And it was one such junior doctor, a guy called Chris Hatton, came to work in my lab. He was extremely keen. The great thing about these studentships is they're not actually part of our projects. So they give us a really great opportunity to investigate something that we just think is interesting. So we took an absolute punt. He started studying a disease that affects babies called Krabbe disease, which I was interested in because heterozygous variants in the gene that cause Krabbe disease are a risk factor for Parkinson's. But in a biallelic state, usually a homozygous state, they cause Krabbe disease, which kills children, sadly, by about the age of 12 months of age. So this is a very rapidly progressive neurodegenerative condition. So we studied alpha-synuclein pathology in the brains of these babies who tragically died, and astonishingly found alpha-synuclein pathology that had prion-like characteristics, which constituted the first report of that phenomenon in the brain of anyone under the age of 60. I think made even more remarkable because the children whose brains we had used were between 10 and 12 months of age. So they were less than 1 year old. One was over one, just over 1 year old. So this was a somewhat chance finding, a totally, you know, kind of out there research project, but it formed the basis of that. And a lot of the work that we've done since, this again comes back to a lot of it's luck. You know, we could never have predicted we would have found what we did. And we found it in another pediatric condition now as well, a related condition called metachromatic leukodystrophy. So yeah, a lot of it is just taking a chance and it happens. How I got to write the thing, Lauren, honestly, I have no idea. I still don't know where the time came from.
So just on the topic of today then, Dan, do you think you can talk about your external network that you've built with collaborators? How did you identify possible collaborators, make that approach?
You're probably going to be unsurprised to hear a huge amount is luck as well. Luck is this word that will keep happening. So probably the most important connection I've made in my career occurred in Fort Lauderdale, Florida, at the International Lewy Body Dementia meeting in 2015. I was invited out for dinner with a few senior folks at Newcastle, and sitting there was a guy called Omar Al-Agnaf, who I'd known by reputation. He's a sort of biomarker specialist from, at that time it was the University of the United Arab Emirates. He then moved to Qatar Biomedical Research Institute, in Qatar obviously. We started chatting. I discussed with him some interesting pathological features that I'd identified in the brains of people with DLB. Particularly we'd found astrocytic pathology, which is very different than the neuronal pathology that's thought to characterize the condition. And he said, I will send you all of my antibodies. So he generates antibodies for biomarker purposes. He sent me all of his antibodies within a month, and we started working together then. I think since then we've published something like 15 papers, most recently in PNAS, where I hasten to add mostly his work, showed that phosphorylation of alpha-synuclein reduces its capacity to aggregate and spread, suggesting that contrary to the common view, it is a protective rather than harmful modification. So this is, I mean, again, I just fell on my feet here because this is a tremendously productive, really well-regarded lab that suddenly I was working with. I was their main point of contact at Newcastle. You know, I did everything a good collaborator should do. I, you know, did the work in a timely manner. I, you know, was very reasonable about, you know, who owns what in terms of academic ownership. And we've just flourished ever since. And I would say Omar is now, you know, a good friend as much as a good collaborator. So, but again, it was luck. And it was a case of literally just chancing my arm and saying, by the way, here's a photo that I'm so sad I have on my phone of something I've seen interesting in brain tissue. And that's what happened. So yeah, maybe sometimes it's about just biting the bullet and doing it. For what it's worth, I always think the most interesting talks at conferences are usually from early career researchers, much more than established academics who are talking about what they did 10 years ago. It's always exciting to hear what's happening at the actual forefront of science. And I think that if you were to approach most academics, they would be really keen to talk to you. I don't think that people should ever feel inhibited because someone's a big name or whatever. Most people are very reasonable.
So have a bit of a shy Burns getting out attitude.
Yes, to use the Newcastle vernacular, absolutely. Shy Burns getting out.
Hey, I'm just wondering, have you ever felt the pressure to move away from Newcastle? You sort of sometimes hear in your academic journey you should work in different labs or different universities. Have you ever felt that pressure?
No, not really. I mean, the reason I haven't, so it's an easy sell for me, because I work on alpha-synuclein, Lewy body dementia, and mitochondria. They're three things that Newcastle is very famous for. So that really helps me a lot. It's a very easy sell for me to say why I'm staying at Newcastle. I've repeatedly said from the start to funders, in interviews for fellowships, whatever else, I've said, I am not going to move simply for the sake of you tick boxing that I have moved somewhere. That is ridiculous. It's anathema to the idea of science, of developing yourself. It's just silly. If something else if another place could offer more than I get now, then yeah, maybe then I'll consider it. But as things stand at the minute, there's nowhere offering me anywhere near what I have at Newcastle. And I don't mean in terms of infrastructure and money. I mean just as I have that as well, but also we have this really, really good working environment where you can't put a price on just cordial working relations, genuinely collaborative and just nice working environment, and that's what makes everyone thrive. So I haven't found it yet, but who knows?
Just going to touch on something else that, granted, isn't directly related to independence, but kind of things that we as more bench scientists are asked to put in our applications, particularly fellowship applications. Patient and public involvement. As
Yeah.
bench scientists, I find that difficult to, I've done a lot of PPI, and I think one of the last sessions that I spoke at, I got asked, what do you want me to contribute to, as a PPI layperson, what do you want me to contribute to science, because I don't feel that I can. Do you have any insight into how we can increase our PPI?
I think it's a really good question. I think it's tricky because PPI is such an important thing in science. It may not always seem, I guess, important, but in some respects, like if you're talking about, I want to look at complex 3 of the electron transport chain, and you're expecting, people who have no science background to comment some insights into that particular project, it may seem, that it's not, something that's that you think is adding much to the project. I think it's important that we as scientists, especially in the translational space, we need to be 100% sure that what we're doing is absolutely aligned with what the stakeholders expect and want. There's no point whatsoever in us doing something because it's interesting if it's not useful to the people who would potentially stand to benefit from it. So PPI is such an important thing no matter what, because you definitely don't want a situation where scientists are doing stuff because it's interesting, not necessarily useful. On the other side, I think it's important that we know what can and cannot be achieved from a PPI perspective. Now, if I wanted a new questionnaire for families about caregiver burden, for example, absolutely PPI involvement is critical. PPI involvement is just different, I think, for bench science. One of the most useful things that I've learned is from our voice group, who actually meet in this building. It's a group of older people from Newcastle, very engaged in research. The most useful thing I found was actually how to communicate with the lay audience. And spoiler alert, whatever you think is lay probably isn't lay. Almost certainly isn't actually. Lay is very, very different. So really useful piece of advice. A fantastic guy called David Innes, who's on our group, and I've stayed in touch with him ever since. He's a retired, I think, history teacher. And he gave me the best advice that I've ever got for writing a lay summary. So remember this. If you remember nothing else I say today, do not include a single word that is more than 2 syllables. And if you do that, you will almost certainly have something that is suitable for a lay audience. Anything that is more than 2 syllables is not appropriate for a lay audience, unless it is a particular term like the name of a disease. It has really helped me a lot. I also understand, and I found this also from a PPI group, that UK government guidelines for any information to be communicated to the general public are that it should be pitched at the understanding of a 9-year-old. That is UK government advice. Now, I've not checked this, but that's what I was told anyway. So always aim for that. For me, I always think of my grandmother, who's 95 and, you know, still going strong. But I always think, could I explain this to her? Is what I'm saying suitable for her, and would she understand it? She has, I don't know, 8 years formal education or something. So that's kind of how I pitch it.
I think we'll say thank you very much. And are you going to be back for a little bit? Or?
Yeah, I'll hang up. Yeah. Thank you.